Have you ever wondered why your hair is straight, but others had voluminous curly hair? Why some people have blue eyes, but you have brown eyes? Or even why you're so short, but others are so tall? Well, these characteristics are being controlled by the nucleic acids in your body. In this video, we're going to explain about nucleic acids. Nucleic acids are long-chain polymers which are made up of large repeating units, known as nucleotides. You can imagine nucleic acids as a chain of pearl or diamond necklaces, with each individual diamond or pearl as the nucleotides. The name nucleic acid comes from the fact that they were first described as acid because they actually had acidic properties, much like the acids that you know. And it is an important class of macromolecules found in all cells and viruses. There are two common types of nucleic acid, which include deoxyribonucleic acid, the DNA, and ribonucleic acid, the RNA. The function of these nucleic acids is to store and express genetic information. The genetic information acts as the instruction to the cell to regulate and to supervise the activity of a cell and the body. This is very important so our body knows what to do at the right time. For instance, our body does not magically hit puberty. It was all controlled by the genetic information stored in nucleic acids. We don't develop secondary sexual characteristics and experience changes in the body too early or too late, but just at the right time. Well, of course, some other factors do influence it too, but nucleic acids, they play the major role. Everyone has their very own unique genetic information inherited from their parents, and thus we are all different from one another. But now you must be wondering, how about identical twins? They look the same. They must share the same genetic material from their parents. Well, it's kind of true that they look the same, and yes, they do come from the same egg. But that does not mean that they are genetically identical by the time they were born. They too, they're unique themselves, and one example is that they have their own fingerprint. Now you're well equipped with what is nucleic acids. Let's see how they look like in detail.
I will now take over and let me be your guide to look at the structures of nucleic acid in detail. Nucleic acids are biopolymers made out of many biomonomers known as nucleotides. A nucleotide consists of three components. The first component is pentose sugar, which is a 5-carbon furanose where the 5th carbon lies outside the ring. The carbons are numbered in a clockwise order, and there are two types of pentose sugar, namely ribose and deoxyribose. The difference is that ribose has an OH group at the second carbon, while deoxyribose has a hydrogen atom at the second carbon. There is no oxygen atom bonded to the second carbon, so it is called deoxyribose. The next component is nitrogenous base. It is a ring structure containing N atoms in varying amounts bonded to other atoms like C, H, and O. There are two types of nitrogenous bases, namely pyrimidine, which is single ring, and purine, a double ring structure. These bases are connected to the pentose sugar at the first carbon via glycosidic bonds. Further information on these bases will be discussed by our next group members. Last but definitely not the least, phosphate group is the third component of a nucleotide. 4 oxygen atoms bonded to a central phosphorus atom, making phosphate group a polar molecule. It has the same structure as in ATP molecules. It is connected to the pentose sugar at the 5th carbon via ester bond. Let me give you an additional information. Nucleosides are basically nucleotides, but they do not have the phosphate group, so do not get confused between these two. And just now I mentioned that nucleotides are monomers, so what do they look like in polymeric forms of polynucleotides or nucleic acid? The phosphate group is the connection site between each nucleotide. It connects them by bonding to the 3rd carbon of a nucleotide and to the 5th carbon of another nucleotide via phosphodiester bond. This sequence repeats and forms the polynucleotide with a sugar phosphate backbone. RNA or DNA? Which one? Which one? The type of nucleic acid form is actually based on what pentose sugar is present. Only one type of pentose sugar can be present in a polynucleotide chain, be it ribose or deoxyribose, never both. RNA contains ribose, while DNA contains deoxyribose.
We have talked about the nitrogenous bases of DNA and RNA, including the purine rings and the pyrimidine ring. Let me talk about more detail. Both purine and pyrimidines are organ compounds that take part in the synthesis of nucleic acid, DNA and RNA. Therefore, they are called the building block of this genetic material. There are two classifications of nitrogenous bases, and we will talk about them one by one. First, let me talk about purine. Any of class of the organic compound of the heteroclastic series, characterized by a 2-ring structure composed of nitrogen and a carbon atom, where a 6-member rings and a 5-member rings combine together. There are in fact many naturally occurring purine. They include the nitrogenous bases in nucleic acid, which are adenine and guanine. Plus, compounds like cysteine, caffeine, and uric acid are also purine. Let's move on. Pyrimidine, another type of heteroclastic nitrogen basis, has only a 6-member nitrogen-counting ring. In nucleic acid, the 3 types of the nuclear bases are pyrimidine, named cytosine, thymine, and uracil. In DNA, purines from hydrogen bonds wear their complementary pyrimidine, adenine with the thymine and guanine with the synthesis, respectively. This is called complementary base pairing. In RNA, the complement of adenine is uracil instead of thymine. In adenine with the thymine or with uracil, 2 hydrogen bonds are made. In guanine with the cytosine, 3 hydrogen bonds are made. Since hydrogen bonds are not strong as covalent bonds, base pairing can easily be separated, allowing for the replication and the translocation. This specific pairing factor into Chargaff's rule, the number of adenine in nucleic acid molecule will always be equal to number of thymine. The same goes for guanine and the synthesis. That's all from the pairing and the preliminary. Now let's move on to DNA.
What is DNA? DNA, also known as deoxyribonucleic acid, is a bunch of atoms stuck together. In the case of DNA, these atoms combine to form the shape of a long spiraling data, sort of like this. If you ever studied biology or watch movies Jurassic Park, you probably heard of the DNA act as a blueprint or recipe for living things. But how? How does this mini molecule act as a blueprint for something as complex as a living organism like the plant, animal, microorganism, viruses, and also us, human? Apart from that, naturally occurring DNA molecule can be circular or linear. The genome of single-cell bacteria and archaea, the prokaryotes, as well as the genome of mitochondria and chloroplasts, certain functional structures within the cell are circular molecules. In addition, some bacteria and archaea have smaller circular DNA molecules called plasmid that typically contain only a few genes. Many plasmids are readily transmitted from one cell to another. In 1953, James D. Watson and Francis H.C. Crick proposed a three-dimensional structure for DNA. Watson and Crick postulated the two strands of polynucleotide coiled around each other, forming a double helix. The two strands through identical run in opposite direction as determined by the orientation of the 5' to 3' phosphodiester bond. The sugar phosphate chains run along the outside of the helix and the bases lie on the inside, where they are linked to complementary bases on the other strand through hydrogen bonds. DNA is a polymer of the 4 nucleotides A, C, G, and T, which are joined through a backbone of alternating phosphate and deoxyribose sugar residue. These nitrogen-containing bases occur in complementary pairs as determined by their ability to form hydrogen bonds between them. A always paired with T through 2 hydrogen bonds, and G always paired with C through 3 hydrogen bonds. The bonding between complementary bases also provides a mechanism for the replication of DNA and the transmission of genetic information. So what about the DNA replication? It's a process by which DNA makes a copy of itself during cell division, which means making more DNA. First, let's talk about where and when. If it's in a eukaryotic cell, it occurs in the nucleus. However, not all cells have a nucleus, such as the prokaryotic cells. They don't have a nucleus. Still, both eukaryotic cells and prokaryotic cells do DNA replication, but there is some difference between both of them. Next, when does DNA replication occur? Well, a cell has to undergo DNA replication before division, so that the new daughter cell can also get a copy of DNA.
DNA replication starts at a certain part called the origin. Usually, this part is identified by certain DNA sequences. Helicase comes in and unwinds the double helix of the DNA. The separation of the two single strands of DNA creates a Y shape called the replication fork. The two separated strands will act as templates for making the new strands of DNA. The DNA strands are anti-parallel. One of the strands is oriented in a 3' to 5' direction. This is the leading strand. The other strand is oriented in a 5' to 3' direction. This is the lagging strand. As a result of their different orientations, the two strands are replicated differently. For the leading strand, the DNA polymerase builds the new strand in the 5' to 3' direction, racing up next to where the unwinding of the double helix. This sort of replication is called continuous. But for the lagging strand, primers have to keep being replaced in order for DNA polymerase to build. Chunks of DNA called Okazaki fragments are then added to the lagging strand, also in a 5' to 3' direction. This type of replication is called discontinuous, as the Okazaki fragments will need to be joined up later and primers will be replaced with DNA bases. At the end of this replication, two identical double helix DNA molecules are formed from one original double helix DNA molecule. The DNA replication is described as semi-conservative, as half of the chain is part of the original DNA molecule, half is brand new. Following replication, the new DNA automatically winds up into a double helix. One last thing. Surely, you have to proofread your work before to catch errors. New strands are proofread to make sure there are no mistakes in the new DNA sequence.
We mentioned earlier that ribonucleic acid RNA is a single-stranded nucleic acid. Usually, RNA has the same nucleotide as the DNA except for the thymine, which is replaced by the uracil. It converts the information from DNA into proteins essential for the working of a cell. RNA is made by copying the base sequence of a section of double-stranded DNA called a gene into a piece of single-stranded nucleic acid. This process called transcription and is catalyzed by an enzyme called RNA polymerase. The main function of RNA is to carry information of amino acid sequence from the genes to where proteins are assembled on ribosomes in the cytoplasm. There are three types of RNA, which are messenger RNA, ribosomal RNA, and transfer RNA. mRNA is a type of RNA that functions by delivering the information encoded in one or more genes from the DNA into the ribosome and passing the instruction about the type of proteins required by the body cells. Therefore, the mRNA plays a vital role during a protein synthesis process. The transfer RNA is held responsible to carry the correct amino acids required by the body to match the correct mRNA codon. This is also known as soluble RNA, and it forms a link between mRNA and the amino acid. The nucleotide sequence is converted into a protein sequence by translating each 3 base sequence, or known as a codon, with a specific protein. So how about the ribosomal RNA? The rRNA is synthesized in the nucleus, and they are the main component of the ribosome. The ribosome consists of large and small subunits of rRNA. Both the subunits combine into one ribosome and move into the cytoplasm of a cell and is used to read the mRNA to form the proteins. You may heard about the central dogma, which is a process of the DNA coding for the RNA through the transcriptions and for the RNA code for the proteins by translation. Well, let's focus on what is the transcription first. In our cell, the DNA safely and stably store genetic material in the nuclei of cells as a reference or template. So transcription is carried out in the nucleus to extract this information. In the state of transcriptions, an enzyme called RNA polymerase will connect complementary bases to DNA. These RNA bases are bonded together to form a single-stranded mRNA.
mRNA is just like a copy you take from a reference book because the copy of information you have is the same as what is stated in the book, but it's not used for long-term storage. Although the mRNA contains the same information, it is not an identical copy of the DNA segment, but is complementary to the DNA template. mRNA carrying this important information can freely exit from the nucleus into the cytoplasm and attach to the ribosome. Now that marks the end of transcript. In the cytoplasm, we have tRNA molecules. As we mentioned before, tRNA carries an amino acid that is complementary to the codon on the mRNA. An amino acid is the monomer of protein. We need amino acid to build our proteins. So it's the tRNA that is going to bring those amino acids together to make that. But how does the tRNA know which amino acids need to bring? This is where mRNA plays its role. It is going to direct the tRNA complementary to the codes on it and therefore determine which amino acids that are being transferred. When tRNA brings in the amino acids, it reads the bases on the mRNA. Do remember, it doesn't read one letter on a time. It reads it in a triplet, which is called a codon. For example, the tRNA will contain a complementary anticodon, UAC. All tRNAs with anticodon UAC will be carrying an amino acid called methionine. A tRNA with this anticodon comes in a pair with the complementary AUG codon, which is a start codon, on the mRNA, and then brings the methionine it carries to the ribosome for protein synthesis. Here is some extra information that we can use a codon chart to determine which amino acid each mRNA codon will code for. tRNA will then leave the ribosome, leaving its amino acid behind. That will be the first amino acid for the polypeptide chain. Then tRNA can go pick up another amino acid that is complementary to the anticodon. This process will keep on going until meet the stop codon. Stop codon do not code for any amino acid, and this is the point where the protein building stops and is complete. Please take note that the process translation is happening at rRNA.
Let's have a brief look at some differences between DNA and RNA, shall we? DNA is double-stranded while RNA is single-stranded. DNA is longer while RNA is shorter. DNA has deoxyribose sugar backbone while RNA has ribose sugar backbone. Next, DNA has thymine base while in RNA, the base is replaced by uracil. Unlike DNA, RNA is found outside the cell nucleus. And lastly, DNA is responsible for storing genetic composition while RNA is responsible for transmitting said composition and helping to synthesize proteins.
Hold on. Do you think that's all about nucleic acids? Definitely not. DNA and RNA are the common nucleic acids. There's one special nucleic acid that people often neglect, and it does not function to store genetic material. Let me present to you the energy molecule adenosine triphosphate, or the ATP. It might be a surprise to some of you, but indeed ATP is a nucleic acid. It is made out of adenine nitrogenous base, a 5-carbon ribose sugar, and 3 phosphate groups. The bonds between the 3 phosphate groups are high energy bonds, and when these bonds break apart, energy is released, which is used up by the cell to do work. Well, that's all from us. Hope you guys had a great time learning about nucleic acids. Till next time.